Welcome. Do our children inherit the impact of our life experiences as well as our genes? That would have seemed a naively crazy question 15 years ago, but today the answer is yes. We now know that if we're stressed, if we eat too much or too little, if we smoke, if we're exposed to toxins, then we can acquire characteristics and pass them down to children and even grandchildren. The idea of epigenetics, as it's called, that parents don't just pass their genes to their children, but subtle differences in the way those genes operate, is one of the fastest growing areas of scientific study. Well, we can't experiment with people in the lab, but we have some accidental experiments that use historical events as a way of studying epigenetic changes. Back in the winter of 1944 to 1945, the Nazis occupied Holland. Running low on resources, the Nazis diverted all of the food to themselves. It's known historically as the Dutch Hunger Winter. Holland went from a rich European country, well-fed, to a situation with less than 1,000 calories a day. 18,000 people died of malnutrition. But what happened to those who were fetuses at the time? What happened was a phenomenon called fetal programming. It turned out that the fetuses during the Dutch Hunger Winter reprogrammed their metabolism in the second and third trimesters. The fetus was sitting in the uterus, and physiologically it was asking, what's the environment like out there? Well, not a lot of nutrition coming in, so it programmed its metabolism to be thrifty, a metabolism designed to store things away. If you give the fetus a little bit of sugar, then for the rest of its existence it doesn't waste it, but it stores it away. If it gets salt, it doesn't throw it away. In the urine, it keeps it. Well, how does this thrifty metabolism cope when confronted with a Western diet? You've got a body trying to be as efficient as possible at storing calories, since there wasn't much food around during your development. Well, that's going to work well until suddenly you have all the food you need and more. Once you have too many calories, or even simply enough calories, your body can't stop storing calories. That's going to cause you to suffer a range of disorders, including obesity and diabetes. What about when pregnant? Well, for a typical amount of food eaten by the mother's going to be atypically good at keeping it for herself. So the baby's going to end up subtly malnourished. Does all this worry you about what life was like for you as a fetus? Anyone who has reproduced or even been a fetus can find this literature unnerving. But perhaps you can also see how adaptive fetal programming could be in nature. The fetus is adapting to conditions in the world it's likely going to grow up in. So the question now is, what is the mechanism that allows physiology to respond to the environment? To help get an answer, here's a revelation from genomics. A revelation that may mean interpreting your personal genome is also going to require that you be able to access your parents' DNA. In late 2009, scientists at deCODE, a company that specializes in analyzing the human genome, reported that in some common diseases, which parent the risk allele came from made a difference. In other words, an allele could predispose someone to type 2 diabetes when inherited from dad, but the same allele actually protected against type 2 diabetes when it was inherited from mum. Well, that told us that male and female genomes are chemically modified in different ways. A lot of other examples have been found where key genes are active or not, depending on whether they're from your mum or dad. And in the jargon of the genetics trade, we say these genes that they are imprinted. Genomic imprinting is an extreme example of epigenetics. Epigenetics, then, is the study of how the activity of genes can be altered without changing the genetic code itself. Epigenetics is from the Greek. It means on top of genetics. It is in addition to genetics. It's not instead of genetics. It's in addition to genetics. In recent years, epigenetics has emerged as a consistent theme in the media in its coverage of genetics. There is a view that a trait has to be determined by nature or by nurture. There is a love of deterministic arguments out there. I think in our culture as well as science, we like to think that everything is learned or everything is inborn. And the fact that it's often a little bit of both just isn't such a good story. Well, epigenetics deals with the age-old question of nature and nurture. And notice that I don't say versus. Scientists gave up the notion of nature and nurture being in conflict decades ago, in spite of that cliche persisting in the public eye. The big insight of epigenetics is that genes are agents of nurture as well as nature. The way epigenetics and imprinting happen is through methylation, the addition of a methyl group, which is a carbon atom plus 3 hydrogen atoms, to particular bases in the DNA sequence. This acts as an instruction to turn a gene off. So epigenetics means that the sequence of the DNA doesn't change, but access to the DNA changes biochemically. And crucially, this altered DNA can then be passed down to children and grandchildren. So a brief molecular biology interlude with this figure. Access to DNA is changed by adding methyl groups to the DNA or by chemically modifying the histone proteins the DNA is wrapped around. You have 6 billion nucleotides in a cell, which together would stretch out for 6 feet. Wrapping the DNA around those histones is evolution's answer as to how you get all that DNA into a tiny little nucleus. If the DNA is methylated, then the DNA gets so tightly wiped up, it's closed for business. None of the proteins required to switch on the gene can get access to the DNA. Dr. O'Brien's told you to think of the sequences of DNA as words in a book. You can change the book's story if you staple or stick some of the pages together. You don't change any of the words here, but you can no longer read certain pages, and that can change the entire story. Well, methylation is like a staple that stops a cell from reading genes. Now, you might be thinking that it sounds like quintessential bad news to shut a whole lot of genes down, but that isn't necessarily so, because gene activity is context dependent. If you're a brain cell, the last thing you want is muscle proteins or liver proteins being made inside you. So you want all the genes that produce these proteins thermally methylated and therefore switched off. What gets methylated where and when will of course be subject to evolution. The prefrontal cortexes of human brains are much less methylated than those of chimps, so there'll be more gene activity in our brains. And this suggests that differences in methylation could be an important clue to human brain evolution. But on the other hand, we also happen to be far more susceptible to cancer than chimps. Cancers typically lose most of the methylation on their DNA, so that all sorts of genes are inappropriately switched on, including genes that tell the cell to divide. Uncontrolled cell division is what makes cancers so dangerous. Cancers involve DNA changes that happen in you. We also have patterns of DNA methylation that pass down through the generations. We've been accustomed to inheritance changing by mutational changes in the DNA sequence, but now we know about this non-DNA inheritance.